And it is my pleasure now to introduce you to Drs. Tone, Petrik, and Lum to share with us how Ovarian Cancer Canada's research progress is redefining ovarian cancer outcomes. Dr. Alicia Tone joined the Ovarian Cancer Canada team as the scientific advisor in May of 2019 and became the OvCAN project manager the following November. Her deep well of experience in the ovarian cancer field includes a PhD in ovarian cancer biology from the University of Toronto, postdoctoral studies in ovarian cancer genomics at BC Cancer Agency, and more than 7 years as scientific associate in the Division of Gynecologic Oncology at the Princess Margaret Cancer Centre. Welcome, Alicia.
Thanks, Steph. So I'm just going to wait for my video to be enabled again, I think, so people can see me. But, oh, there you go. Thank you. So I am very, very happy to present highlights of the great progress we have made in research at OCC. For this session, I'll provide a brief overview of research either funded by or conducted by OCC, followed by short presentations by two preeminent Canadian scientists, Dr. Jim Petrik and Dr. Julian Lum. I'll then share the central role that research will play at OCC going forward, with hopefully some time left for some live Q&A. So as many of you already know, a large part of research at OCC is OvCAN, our $14 million plus national initiative aimed at identifying novel treatments for women living with ovarian cancer. This initiative consists of 3 scientific priorities co-developed with the research community, including priority 1, investing in high-quality research models of all types of ovarian cancer. And this is to provide the necessary scientific toolbox for all Canadian researchers to discover potential new treatments. Priority 2, which is testing the impact of new treatments in these research models in preclinical studies. And priority 3, early phase clinical trials using a personalized medicine approach where treatment is tailored to the specific properties of a patient's tumor. So what have we accomplished now that we're in the final year of OvCAN? Just some highlights. So we have 25 projects on the development and or characterization of shareable gold standard ovarian cancer research models to fuel discoveries. We have 13 preclinical studies, including 5 on novel immune-based treatments, 5 on targeted inhibitors, and 3 novel gene targets. We have 250 researchers and clinicians in our OCC Research Consortium. We have more than 20 patient partners in research representing 7 provinces. We've funded 5 interventional clinical trials and 1 pragmatic clinical trial, also referred to as a real-world clinical trial. We've funded 75 plus research personnel and 29 principal investigators throughout Canada. We're also funding 7 tissue banks in 6 provinces, and all main types of ovarian cancer are represented. So a key component of OvCAN, and actually all research at OCC, is the Patient Partners in Research program. Developed in 2020 by OCC, the role of the PPI program is to train and match patient partners to research opportunities to keep the voices of those with lived experience at the forefront of research. And this is to complement and maximize the impact of research being done by Canadian researchers. The Patient Partner Program is co-led and managed by 2 OCC research staff and 2 patient advocates. It includes a diverse representation of ovarian cancer types, age, sexuality, cultural backgrounds, and geography. Some examples of activities that our team members have been involved in are highlighted here. So they have served as patient reviewers on grant funding and trainee review panels. They have been speakers at internal and external events. They have shared their experiences and learnings with both research teams through Bridging the Gap sessions and clinical trainees. They're embedded as research team members on OCC-funded grants, including both clinical and preclinical projects. They've also consulted on research study design, funding applications, patient decision aids and education tools, OCC strategic planning, their own program guidelines, as well as patient engagement best practices. They also stand alongside OCC and the research community in research and system advocacy. So one thing I want to emphasize is that research at OCC goes beyond OvCAN. With a total of 67 research projects in our portfolio, we are driving progress in different ways. These include as funder, collaborator, leader, and convener. We also fund and/or conduct research along the ovarian cancer continuum. This includes, for instance, treatment, prevention, early detection, and survivorship. We also fund projects on understanding the tumor biology of the different types of ovarian cancer, which will feed into future projects in all of these areas. One area of focus for OCC-led research is understanding the ovarian cancer experience from both the patient and clinical perspective. So the Every Woman Study, Canadian edition, which we published in 2022, summarized findings from our comprehensive survey of 557 individuals living with ovarian cancer from across Canada. This revealed inequities in the time or pathway to diagnosis, treatments received, and access to genetic testing and clinical trials. We're currently planning for the second iteration of this study with the goal of improving diversity of the participants. We also have the State of the Nation clinical audit, and this is using administrative data from 5 provinces to determine ovarian cancer incidence, prevalence, staging, and survival. We're also proud to lead the National Ovarian Cancer Prevention Task Force, through which we are collaborating with scientists, gynecologic oncologists, genetic counselors, menopause specialists, including Michelle Jacobson, family physicians, patients, and previvors, or those who are at an increased risk for ovarian cancer, to identify and address important gaps in the prevention pathway. OCC will bring the voice of those with lived experience to pre-selected research projects and will mobilize the knowledge gained to educate the patient and previvor communities and identify advocacy priorities. So I'm now going to turn this over to our guest speakers. First, I'll introduce Dr. Jim Petrik, and then after him, I will come in and introduce Dr. Julian Lum. So first up, Dr. Jim Petrik from the University of Guelph. He is a part of a collaborative research program to develop novel therapies for advanced stage ovarian cancer. And he is an expert on tumor blood vessel formation and how the manipulation of tumor vasculature is an important component to anti-cancer therapies. So over to you, Jim.
Thanks so much, Alicia. It's a fantastic honor to be here. I always love participating in Ovarian Cancer Canada events, and I'm really very happy to be here. Can you see my slides and hear me okay, Alicia? Perfect. So as Alicia mentioned, much of the work in our lab is designed to look at novel therapeutic opportunities in the advanced cancer space, which is kind of our unfortunate current clinical picture. Our lab is interested in looking at therapies that may help to induce regression and hopefully elimination of disease in the advanced stage. As Alicia mentioned, one of the things that we're really interested in is looking how blood vessels are involved in the tumor development process. Tumors, in order for them to grow beyond 2 millimeters in size, they actually are required to recruit and develop new blood vessels, and the tumors inherently understand this. So one of the early events in tumor formation and progression is this initiation of blood vessel formation through a process called angiogenesis. So it's a big term that reflects a secretion of these factors from the tumor that stimulates the nearby blood vessels to to sprout and develop blood vessels that then migrate towards the tumor and invade the tumor. And theoretically, they're designed to bring blood supply, oxygen, nutrients, et cetera, to the tumor to facilitate growth. So the tumors understand this, and conceptually, it makes sense that they would want to recruit their blood supply. But what actually happens is the tumors secrete such an aggressive blood vessel formation program that the blood vessels form so rapidly that they're actually very poorly formed. And as you can see in the bottom of the slide here, hopefully you can see my pointer. Normal blood vessels are very well organized. There's a nice roadway of vessels, and they're very efficient at bringing a blood supply into the tumor. We see with tumor blood vessels, because of that very aggressive angiogenic program, that the blood vessels are formed very atypically, they're dysmorphic, and they're actually very, very inefficient. So we end up with a situation within the tumor where the environment within the tumor is very hostile. So we find that because of that dysfunctional, abnormal tumor blood vessels, we end up with areas that have very little oxygen, and leads to a term called hypoxia. Because the blood vessels are very leaky, a lot of fluid leaks out. So we end up with a tumor that's very highly pressurized. There's a lot of fluid that accumulates inside the tumor. So if you think of it from a therapeutic perspective, most women that undergo chemotherapy rely on intravenous delivery of chemotherapy drugs or other therapies to the tumor. So we've already talked about the supply is very dysfunctional. The tumor is very poorly perfused because the blood vessels aren't formed very well. And then because of the leakage of fluid, we know that there's a lot of pressure inside the tumor. So we're relying on a dysfunctional supply system to get therapies into a very highly pressurized tumor. So conceptually, it makes sense that therapy uptake into the tumors is compromised. And that's one of the reasons why therapeutic efficacy has had limited kind of success in advanced stage cancers, because we can't get the compounds where they need to be. So I've had a long-standing, wonderful collaborative career with Dr. Jack Lawler from Harvard, and I don't want to make this complex at all, but we've developed an approach where we've identified this protein called thrombospondin 1, but we've identified a very small region of that protein that's incredibly potent in remodeling the tumor microenvironment and affecting the tumor vasculature. We know that it inhibits some of those factors that promote blood supply to the tumor. And our initial thought was, we're going to treat with the 3-TSR, or the type 1 repeats, which is our molecule, and we're going to kill all the blood vessels to the tumors. We're going to starve the tumor, and the tumor is going to implode and go away. And we thought this is potentially really fantastic. And this is an example of the data that we got in our first run. And you can see in the blue are tumor cells. So the blue outlines the tumor. You can see in red are the tumor vessels. And hopefully you can appreciate this really kind of ridiculously unorganized tumor vasculature, large amorphic blood vessels in the control group. And in the treatment group where we treated 3-TSR, we thought we're going to see a complete absence of blood vessels, and we're going to have a starved tumor, and it was going to go away. Well, 100% the opposite occurred. We saw blood vessels that were beautifully formed, very functional, small tubular luminal blood vessels that were matured with the presence of these green cells, which are smooth muscle cells that line really nice functional blood vessels. And we thought, well, what the hell? We have no idea how to deal with this. This was completely contrary to our hypothesis. And so I had no idea really how to reconcile the results from this. And this was completely against what we were hypothesizing. So I really struggled with this for a long time. And I thought, well, is my career over? I have no idea what to do. So we identified then a potential therapeutic opportunity. We realized that when the tumor blood vessels were now normalized through this vascular normalization process, well, perhaps if we've made blood vessels more functional, maybe we can exploit that. And we looked at the level of tumor hypoxia, or the amount of tissue that is poorly perfused was way down. So we thought, well, let's test out the hypothesis that maybe these good blood vessels may be functional and may allow us to improve delivery and uptake to the tumor. So we did a simple experiment where we made chemotherapy drugs radioactive, and we infused either control animals or ones that we treated, and we saw that there was a dramatic increase in the uptake of the therapeutic compounds in the tumor. So this was fantastic. This was kind of, you know, my eureka moment in my career, where we saw that we may be able to enhance the uptake and efficacy of these therapeutic modalities into an environment that historically is very refractory and very inhospitable to therapies. So we wanted to look at, can we take advantage of this enhanced delivery? And we know that in cancer therapy, most individuals, most women, ovarian cancer patients undergo maximum tolerated dose chemotherapy, where they are subjected to high levels of chemotherapy, undergo a drug-free program to recover, and then go through more cycles of chemotherapy. This is problematic for a couple reasons. One is that, as everyone here, unfortunately, is very well aware of, the high level of chemotherapy is very difficult to deal with from side effects, and it's so debilitating. But the other problem is, during this drug-free interval, this is the opportunity that the remaining tumor cells that weren't killed off, provides them the opportunity to develop their chemoresistance mechanisms. And we see kind of repetitive development of chemoresistance through these selective maximum tolerated dose scheduling. So there's a different type of chemotherapy that's out there now called metronomic scheduling, where individuals will undergo more chronic dosing with very low levels of chemotherapy, which has the advantage of being less difficult from side effect perspective because there's much lower dosages. But the other benefit is that there's no longer these drug-free intervals. But the problem is, if we're only going to give small amounts, that's challenging, because we know that it's hard to get enough drug into the tumors, and that's why we give such high doses in the MTD scheduling. But we thought, look, if we've enhanced delivery to the tumors, maybe we can manipulate and optimize the uptake and allow metronomic chemotherapy to have an impact. I know this is a busy slide, but I'd like to direct you to, here we see, the control animals that have large tumors. The rest of the treatment groups, the maximum tolerated dose, metronomic chemotherapy. But if you look at the combination of the normalized tumors with 3-TSR along with the metronomic chemotherapy, we saw that there was a dramatic reduction in tumor size. And when we looked at overall survival, at 150 days post-treatment, which is half of a mouse's lifespan, we had only lost one mouse in the entire cohort. So we were really excited about those results. We're now working on another compound that's an improved compound that really increases the clinical potential for our therapeutic approach. We developed a new compound that has a lot longer time in circulation, so from 14 hours to 8 days, which means that a woman would only have to undergo infusion once a week as opposed to daily, which obviously isn't really clinically relevant. So we're bringing this forward. We're working with Health Canada right now towards a clinical trial with this compound. But one of the things that we're really excited about, we already showed that some data to here tonight that we can enhance delivery to the tumor. But the other side of it, if you remember, within the tumor, they're very highly pressurized structures, and that's a main impediment to therapy uptake. So we looked at what does the pressure look like when we remodel the vasculature and normalize the environment. And we see that in response to our therapy, the pressure within the tumor drops precipitously. So that creates an environment now that is more likely to be hospitable to therapeutic uptakes of any kind. We've looked at chemotherapy drugs, but I can tell you we've looked at immunotherapies, checkpoint inhibitors, oncolytic viruses, adoptively transferred T cells. And we can see that we get about a 500% increase in the uptake of any of the therapies, whether it's immunotherapies or chemotherapies or viruses. So we're really excited about that. One of the things this is just a cool picture to me. I know it's kind of a science nerd picture, but to me, this is so exciting. One of the problems with having a highly pressurized tumor is that it collapses the very fragile lymphatic blood vessels. Now, the lymphatic blood vessels are the vessels that drain the tumor, so drain fluid from the tumor. But not only are those vessels really important for creating a conduit for fluid drainage, but that is the pathway that immune cells, activated immune cells, take to go to the draining lymph nodes to educate the peripheral immune system that the tumor cells are a bad thing. So if we can get activated and educated immune cells to go from the tumor to the draining lymph nodes, then all of the immune cells in the body become educated that tumor cells are bad, and we can have whole systemic immune response to the tumors. And so we did an experiment, and this is just, to me, such an exciting image of lymph nodes. So this is lymph nodes here of the control group, and these are lymph nodes of our treated group where we stained for the presence of immune cells. And in green and blue and red, we see different types of immune cells, which isn't really relevant. But I think the take-home message here, the really exciting take-home message here for us, is that we see such a dramatic increase in the migration of these activated immune cells that go from the tumor to the draining lymph node. And now this animal's peripheral immune system now is educated against the tumor. And with this modality, we're able to start treatment in the advanced stage space. We're able to allow animals to get sick to about a stage 3 ovarian cancer phenotype. We initiate treatment at this stage, and we're able to induce complete remission and eradication of metastatic disease, because we think that we're facilitating uptake and efficacy of these therapies, whether it's, once again, chemotherapies or immunotherapies or oncolytic viruses. So really our therapeutic benefit is, it has a benefit on its own. We know that 3-TSR, FC3-TSR has a lot of benefit on its own, but its real, I think, impact will be when we combine it with therapies. We know that immunotherapies haven't been as successful as we'd hoped in ovarian cancer, and we're very hopeful that combining it with our therapeutic approach may really revitalize the therapeutic impact of some of these therapies, like checkpoint inhibitors and PARP inhibitors and things like that, because we can get the compounds and the relevant players like immune cells into the places where they need to be. So just to take in kind of a way of summary, to take a bit of a graphical depiction, if we look at the dysfunctional tumor vasculature, we're impeding flow, we're impeding delivery into the tumors, we're impeding deliveries to the draining lymph nodes, we're not allowing immune cells, we're not allowing drugs to get where they need to go. Where when we remodel the microenvironment, we open the highway back up, and we allow things to get where they need to go, and we're really excited about that. And, as a PI, I have to thank all the people that did the work, but I just want to tell you, I know I have a second left before Alicia's people come and bust my kneecaps, because I'll be over time. But one thing I do want to really recognize is the contributions that Ovarian Cancer Canada has made. Much of the work that I've showed today has been at least partly funded by Ovarian Cancer Canada through a number of grants that I've received over the past number of years. So, I'm greatly indebted to this community here for, first of all, inspiration. I go to a number of the patient partner groups and survivor groups, and I always take my grad students, and that's been such an inspirational interaction and engagement. And I'm grateful for Alicia and OCC for facilitating that. But I'm also extremely grateful for the research funding that's really catalyzed some of the work that I was able to present today. So I really appreciate the opportunity to speak at the symposium here. It's an honor to do so, and I know there'll be an opportunity for questions later, I think, so I'll be happy to take some then, but thank you very much for your time and your attention.
Thank you so much, Jim. And yes, there will be time at the end for questions for both you and Dr. Lum. So now I'm going to introduce Dr. Julian Lum from the University of Victoria. Dr. Lum's current research interest is focused on the relationship between metabolism and immune responses in ovarian cancer. The broader goal of his lab is to identify potential metabolic targets that can be used to enhance cell-based immunotherapy. He's also working very actively, actually, with our patient partners and research team to design a brand new program on nutrition in ovarian cancer. So take it away, Dr. Lum.
Great, thank you, Alicia. I'm just trying to start my video. There we go. Excellent. And then I will also share my screen. Just if you could give me a thumbs up if that's all coming through. Fantastic. All right. Well, thank you, everyone, for joining this evening. It's getting late on the East Coast. We're on the West Coast here, so we're still a bit earlier than you. But thank you for coming in. It's really a pleasure and a great honor to be here. And I'd like to start off with a territorial acknowledgement. My lab and my team, we work on the traditional territories of the Lekwungen people, specifically the Songhees and Esquimalt First Nations. And we acknowledge and respect our traditional hosts. We thank them for allowing us to conduct the research on their lands. And we've taken sort of our approach to reconciliation a bit further in my team. And one of the things that we've been doing is trying to keep everyone sort of up to date on efforts around the globe, locally, etc. And I just wanted to point this one fact out that has transpired recently. So the Aboriginal and Torres Strait Island people just make up a little less than 4% of Australia's population, and they've inhabited Australia for over 60,000 years, but they're not mentioned in the Constitution in any way, and they are the most disadvantaged peoples by most socioeconomic measures. So despite accounting for almost 4% of population, there's really a disproportionate socioeconomical disadvantage from suicide rates, the average lifespan, and disproportionate diseases that are found in some of the outback remote areas that have already been eradicated in other parts of Australia. Several weeks ago, I didn't realize this, but Australia can institute a mandatory vote, so all citizens must vote. But there was a landmark referendum where more than 60% of Australians voted no when asked whether they should alter the Constitution to recognize Aboriginal and Torres Strait Island peoples. And so I just brought this up because I wanted to let people know and be informed about what we're doing and what we continue to do to sort of perpetuate this disparity and in our own reconciliation processes wherever you live. Okay, so with that, thank you, Jim, for your presentation, because I think it segues very nicely into the topic that I'm going to speak to today. And really, I wanted to take a big step back and look at ovarian cancer really from a very high level. And I kind of use this analogy of a planetary view of this disease. And, to me, I think, as Jim has alluded to, we've tried for decades with a number of different approaches, both all the way from, early diagnosis and therapeutic treatments, etc. And we have made some strides, and we need to make more strides. And how can we make more inroads is really the question that I've grappled myself with over the past few years. And so I'm going to ask the question to the audience, and you can cogitate about this, on the call here. But what is cancer, and what causes ovarian cancer specifically? And this is a really important question from the standpoint of, if we know the driving forces, can we then design therapeutics or strategies that get to the root cause of the problem, as opposed to thinking about more of the distal features of cancer that arise later on in disease? Can we get to the trunk, essentially, of the tree, the roots of the tree? So this illustration is what is called the hallmarks of cancer. This is sort of the major features that have been described as the reasons for why cancers emerge. And I won't go through all of these details, but some things like avoiding immune destruction, resisting death, genomic instability, all of these things. And Jim touched on angiogenesis, the vasculature. And these are all problems that we have known for many decades as the original hallmarks. Some enabling features in purple, and then some of the new and emerging features and hallmarks of cancer as a broad class of disease. And so another question I'll ask you tonight is, what do all of these all of these features have in common? And I think that's a very important point. Again, getting back to this concept of like finding the root cause of ovarian cancer. And we've been thinking about this a lot. And one of the key features is that all of these cellular processes that I've just flashed on in the previous slide, they require energy. And I like to use the analogy clean energy. And what does that mean? They need an ability to generate the energies to fuel all of these processes. So they need proper metabolism, they need proper nutrition, and proper diet, right? So the cells need all of these things in order to be able to drive and fuel these processes. So at the root of all these complex processes that are happening inside the cell is the innate ability and use of the foods that the cells can acquire from the environment. And so we have begun to develop a program around how can we use diet and metabolism as a way to promote immune health, because immune health is one of the main pillars of how cancer cells evade the immune system and develop into full-blown disease. And you know, our goal is really to improve the immune response to cancer so that we can have better outcomes. Okay. And so you might be asking the question, well, how do we get to this point? How do we achieve this goal? And I think, you know, keeping on this theme of metabolism, it's really about inclusive and sustainable foods. How do we fuel our immune system so that the immune system can recognize and destroy the cancer? And so what do we need to consider in this equation is the question. And as you can imagine, food is intimately linked to our families, our behaviors, our social upbringing. There are economic barriers related to access and to food. And so we will really want to develop a very evidence-based, science-based approach to learning about what are the things, what are the foods, what are the nutrients that will drive these improvements in our immune system. So it's really creating a team, I think, that can help us achieve this goal. And the team really is us, all of us here on this call, patients, patient families, caregivers that come from all walks of life that are in this journey together. And so using this as the backdrop, I think it's really important that whatever type of research that we embark on related to metabolism and diet really is inclusive, but also brings together the expertise that we need. So I'm going to tell you about a project in a few moments, but where we have brought together immunologists, we have brought together dietitians, oncologists. We're very interested in changes in the gut microbiome. So these are the healthy bacteria that live in your gut, and we've brought on specialists in that area. We need to talk about and really engage in food producers, the people that will provide the clean energy, if you will. Metabolism experts, biostatisticians, and psychology experts to help us understand, behavioral issues related to food. Indigenous land stewards is a very important component. And then throughout this, I think ultimately, as I showed in the previous slide, our goal is to get this to the patient. And so intricate and integral to what we want to create is an engagement process that has patient partners that will help inform what are the best practices, what are the ways that we can deliver on our goals. And some of you who have been involved in different patient engagement activities and initiatives at Ovarian Cancer Canada may have come across some of these different models. So there's a paternalism model where we have experts who discuss and make decisions, and those decisions and outcomes are essentially transferred to the patient, where as you can tell from this pictorial, there's very limited intersection and engagement. And then there's a patient-centered approach where there's back and forth from patient to experts, and I think this has been a model that has been adopted, but we're moving towards what we like to call partnership of care, where we start with the patient view and the patient perspective, and they're the caregivers, if you will. They help to inform the decisions that the experts might be using in terms of practice changing clinical trials, etc. And they are critical members of our care team, and they provide very important life project focus to what we do. And this really is built on a collaborative co-leadership model. Okay, so with the last few minutes of my time, I would like to introduce a study that we have been developing over the last 6 months with Ovarian Cancer Canada. And I think this really speaks to the points that Jim brought up about the tumor vasculature and starving cancer cells and improving immune responses. And it's all very intricately related to what we as organisms, as humans, consume in our diet. And so this study is really designed to ask 2 questions. The first question is, how do different nutrients in our food impact our immune response to ovarian cancer? And the secondary question of this is, how does this involve our gut microbiome, the healthy bacteria in our gut? And I'm not going to go through the laboratory data. Suffice it to say that we have discovered unexpectedly, so like Jim alluded to, these are the real gems in science, because when things don't go the way that you expect, it really informs something new and potentially groundbreaking. And in the laboratory, we have found that if you reduce a certain type of nutrient, and this nutrient is called methionine, in your diet, they tend to improve the immune responses in the laboratory. And so we are designing a study where we're going to limit the amount of methionine in patients. We're going to collect different samples over the time of this study. And we're going to study the immune response, and we're also going to study the gut microbiome. And so we've engaged the patient partner research group to help us create this study. And I've noticed some names on the line tonight, and I want to thank you for being part of this study. And I think there are a lot of important questions. I won't go through all of them, but these are some of them that I think having the lens of the patient partner really will help us design a study that will not only be successful, but will inform practice changes, we hope. So what is the right way to message a diet? What is the best patient group in terms of the trajectory of their disease? What are the best practices in terms of tracking information electronically, etc.? And because this is a diet study, what are the different ways that we need to think about cultural food practices? That's very important. How do we deliver the food, these diets? We have different modalities, and again, I won't go through the specific details as we're still developing this alongside of the patient partner research team. But we have things like a choose your own adventure. We have cookbook style recipes. We're even thinking about a full delivery service using, something like a HelloFresh or a local company to help us with. So I'm going to stop here. Again, I want to thank Ovarian Cancer Canada for this partnership and really allowing us to engage the patient group, and we're hoping that this will really create a study where it's designed for the patient, it has the patient lens in mind, and we hope that this will lead to really changes in immune responses and better outcomes for patients. So thank you for your time tonight, and I'll stop here.
Thank you so much, Julian. It's really been amazing to watch the progression of the study as it's developed and really see the patient partners' involvement. So again, thank you so much to Dr. Petrik and Dr. Lum for such exciting presentations. To close out the session, I wanted to give a brief overview of how we're keeping the research momentum going. So we are now in the final 6 months of OvCAN, but our research efforts are not stopping. We've already developed a comprehensive research plan up to 2029, which is laser focused on funding research that will impact outcomes along the entire ovarian cancer continuum, from prevention all the way to survivorship. The patient voice will continue to be at the center of all research activities, and we will be increasing our investment in the next generation of ovarian cancer researchers, as well as understanding the impact of social determinants of health on the ovarian cancer experience. This comprehensive research plan has been incorporated into OCC's new strategic plan. To highlight some of the strategic priorities that are especially relevant to research, we want to improve patient outcomes through scientific discoveries and research breakthroughs. We also want to elevate the patient voice. We want to elevate the voice of the ovarian cancer community. These as well as all the other strategic directions, feed into our winning aspiration to be the ovarian cancer research engine that sustainably drives health outcomes, influences system changes, and improves the quality of life for women with ovarian cancer and those at high risk of the disease. I will also want to emphasize the interconnected roles of research, advocacy, and knowledge mobilization in achieving our mission. So as mentioned, we have research that is laser focused on changing outcomes along the entire ovarian cancer continuum. These findings then inform priorities for advocacy as well as knowledge mobilization. And by advocacy, I'm referring to multiple things, including federal and provincial research funding advocacy, as well as health system changes. In terms of knowledge mobilization, we're going to use these research findings, especially those that are enhanced by having the patient or previvor voice, into our resource guides, self-advocacy tools, national education and research symposiums such as this, and speaker series on special topics. So with that, I'm going to end the formal part of the session and open it up to Stephanie to moderate the Q&A. But first, I just want to thank our funders, funding partners, clinical and scientific collaborators, and especially the patient partners I have the honor of working with. Thank you.
Thank you, Alicia and Dr. Lum and Dr. Petrik. We have just a couple minutes for questions, so I'm going to get right to them. Dr. Petrik, I have a couple for you, and then Dr. Lum, I have one for you. So, Dr. Petrik, how close are you to clinical trials?
It's a
$1 million question.
Yeah, for sure. So where we are, we've made a lot of progress. We've done a healthy, a safety study in a cohort of healthy companion animals. We're now doing some pharmacokinetic studies, and we're engaging with Health Canada right now for approvals. So we hope within a year that we'll be going to phase 1 clinical trial. That's our hope. There's some hurdles and a little bit of financial hurdles to get to that next level, but we were already have a partnership to undergo production in a GMP facility. So we're making pharmaceutical grade compound now. So we're excited. We're really excited.
Awesome. I can see everyone on the line is super excited about that too. So Dr. Petrik, another one for you. You talked about tumor uptake, but is this manipulation work on patients that have had their tumors removed?
Yeah, absolutely. So, it was kind of a short kind of presentation. All of the things that we showed are applicable to the primary tumor, but that's usually a surgeon's problem, as I think you're kind of suggesting. But the exact same mechanisms occur in the metastatic tumors, and that's why we're able to see eradication of metastatic disease. Is that those metastatic tumors that infiltrate throughout the peritoneum are also subject to this hostile tumor microenvironment. So we're able to remodel the tumor microenvironment in those metastatic tumors, make them susceptible to the therapeutic intervention, and induce resolution of that widespread disease, which I think is really the exciting thing for me, because we know that the metastatic disease is really the clinical problem or clinical challenge.
Peggy Pickett just wrote a comment in here, and I wrote the same thing down here. I got goosebumps when you said eradication of metastatic disease.
Yeah, and I always hesitate to overstate where we are, but those are the data that we've seen this is all preclinical models, and we know that we have a leap to go to human patients. We understand that, we're respectful of that, but what we've seen so far is really exciting.
Hey, Dr. Lum, I'm going to turn it over to you. I have the questions keep rolling in, so I've got about 3 minutes left. Is CAR T therapy being looked at for ovarian cancer? And are there clinical trials being done in this area?
Yeah, thank you very much for that question. And certainly there is activity. At the moment in Canada, there is activity around using some new CAR T therapy approaches through BC Cancer here in Victoria. As some of you may know, we have a program where we can manufacture these CAR T cells that can be then given to patients. And so we're still in the early stages of developing that trial and what the trial will look like. But I think there's a lot of excitement, and I would just ask people to stay tuned, because this will be something that will be coming online fairly soon, we hope.
We'll be watching. We have 3 or 4 questions here about this. Could you give an example of the low methanion diet?
Yeah, thank you for the question. So a low methionine diet is equivalent to someone who would essentially be a vegan. And the way that the diet is set up is that because you're limiting the source of the methionine, you're also going to limit other nutrients that are related to methionine, which is a class of nutrients we call amino acids that are found in protein-based foods. And so there's a drink that will allow you to maintain your normal levels of those other classes of amino acids, but still be low in methionine. And, it's a 21-day study. And so it is really for a short period of time. And I think that, this is why the patient engagement is so critical, because we need to understand, for someone who is, normally not used to such a vegan-like diet, what does that look like for them, and how can we help them and facilitate and ensure that, that they can actually go through the study from start to finish. So great question.
Awesome. I know we're out of time, but I have one more, because I think this is an interesting question right at the very end. I donated my tumor. Can you talk about how donated tissue is used?
Fantastic question. And, we use the tumors that have been given to, us researchers in multiple ways. We study the genomics, so we want to understand the makeup of the cancer. We can use those as part of the Ovarian Cancer Canada research programs to create these avatars, right? These 3D models that we can test, like Dr. Petrik's, strategies of drug delivery. We can test then our immune cells able to attack and kill the cancer cells, and if so, why aren't they? And we can do this all in a miniaturized fashion in the laboratory, in the Petri dish. So the tumors are such an invaluable resource to what we do on a daily basis. And I want to thank anyone and everyone who, signed a consent form that allows their specimens to be used for research, because it's really propelled, what we've learned and what we can continue to learn.
Thank you for that. Sometimes it's nice to hear about the basics as well. So thank you. As you've seen kind of tonight, we put the patient at the forefront of everything we do. You heard patient experience in all of the sessions we did tonight. And I loved hearing from each and every one of you on this session right now, how critical patients are to the work that you do. So thank you. I want to again thank you for taking the time to spend with us tonight. You obviously are all very passionate about your work, and it comes through in your presentations. I know and I'm certainly everyone on the call, appreciate you putting it in language that we can understand and make us excited about. So thank you all for that. The research that is happening in this community is changing outcomes. I think Alicia had a point on her screen where it's laser focused on changing outcomes. So that brings us to a close of the first night of our national symposium. And once again, I would like to thank our sponsors for the evening, Merck and AstraZeneca, for helping to make this event a reality. I also want to extend my sincere thanks to all of our speakers this evening who shared their expertise and their lived experience with us. And a huge thank you to you, the audience, for tuning in tonight.